Bismillahirrahmanirrahim. I am Dr. Anwar Hussain, HOD Anatomy, FMU. I welcome you all first year class in the e-learning program Anatomy Department, Faisalabad Medical University web page. I want to continue my lecture through this program, and today lecture is about cytoskeleton. You all know that the cytoskeleton is the system of tubular network present within the cytoplasm. As the name indicates, consists of two words, cyto and skeleton. Now, cyto mean cell, skeleton mean framework. So, it is the network of tubular system which present within the cytoplasm and connect the cell with the exterior as well as within the cellular organelle from the nucleus to the plasma membrane. In front of you, the slide is about the types of cytoskeleton elements. One is the intermediate filament, in between is the microtubule, and lower one is the actin filament. This diagram will tell you how this cytoskeleton is interconnected throughout the cytoplasm with the organelle, with the nuclear membrane, and with the plasma membrane as well. Now, what is the introduction? It is a complex network of interlinking filaments and tubules that extend throughout the cytoplasm from the nucleus to the plasma membrane. So, it is understandable starting from the nuclear membrane up to the plasma membrane of the cell throughout the cytoplasm. Eukaryotic cell contain three types of cytoskeleton filament: microfilament, microtubules, and intermediate filament. What are the functions they perform? Number one, give the cell its shape, mechanical resistant to deformation, with extracellular corrective tissue and other cells to stabilize entire tissue. This function is important function because it gives the shape. Shape means the shape is definitely variable from cell to cell. Some cells are round, some cuboidal, some columnar. So all these shapes are maintained by the network of these tubules. Moreover, the cell also stabilize with the surroundings, with the other cell as well, because the cells lies in combination within a tissue to perform a specific function. It also, second function is, it's involved in many cell signaling pathway in the uptake of extracellular material, endocytosis, segregate chromosomal segregation during cell division. This is a very important function of a microtubule and microfilament. Endocytosis is the process through which the foreign material from the exterior is engulfed inside the cytoplasm, and then it is moved properly to the proper place in the cytoplasm with the help of all these microtubules. The chromosome movements during cell cleavage is important function. During cell division, you see that the chromosome has a well defined round structure, that is the centromere, and that centromere is attached with the chromosome microtubules, and these microtubules then pull apart the diploid number of chromosomes to move within the nucleus of the newly daughter cell. The last function is it forms a specialized structure, especially in the epithelium, You see the flagella for movements, cilia also for movement, especially in the respiratory passages, lamellipodia and podosome. These are the structure which are concerned for the anchoring filaments within the nucleus and the plasma membrane. Now, this slide will tell you the all structure components as well as the function of the various macrotubules. Now, on this you see a columnar cell. On the left side you see a columnar cell having a microvilli. Now, these microvilli are also the components of the macrotubule, and these are horizontally running fibers beneath the apical cytoplasm of the cell, and this is made up of actin filament. This is the cytoplasmic organelle, and these vertically running macrotubules, Golgi apparatus, up to the nuclear membrane. No, this is all types of variety: macrotubule, intermediate filament, microtubule, motor protein. So this is the structure of a cell which is going to be divided into two. Now this is the nuclear component, chromosomal component on which attach the macrotubule, and these macrotubule pull the chromosome toward the daughter cell. This is again an example of the intermediate filament in the specialized organization present in the contractility and motor fibers. Now, brief introduction about the actin filament. Microfilaments are polymers of the protein actins, are 7 nanometer in diameter. Now, what is actin filament? These are made up of protein which is known as actin protein. The diameter of this protein is 7 nanometer. Macrofilament composed of linear polymers of G-actin protein. They will organize in such a way that one end becomes, which is also called plus N, is the growing end, whereas the negative end is a non-growing end. And growing end have the multiple unit of G protein which join end to end to elongate the actin filament. These subunits then assemble into two chains that intertwine into form a called an F-actin chain. Now, two chains, they will then twist and bind each other, forming a twisted fashion of F-actin chain. So, double helix F-actin chain will be present within the cell. Function include the muscle contraction. Now, in the histology, you know very well that is the muscles of three types: that the cardiac, smooth, and skeletal. The muscle protein, that is actin filament, form the thin filaments, and they participate in the light band of the cross striation with the visible within the muscle. Whereas the myosin, which form the dark band, is the myosin protein. Now, actin filament, they are present and attached in the sarcomere, attached with the Z line. So they form the light band, and they are stationary protein, whereas the myosin is a protein which moves slide inside to show contraction or relaxation of the muscle. Cell movements, quite obvious because these actin filaments play a key role in the movement of cells, especially if you remember the movement of pseudopodia. Now this pseudopodia having present within the amoeba, so this pseudopodic movements in amoeba are due to the actin filament. Intracellular transport and trafficking, again example of this very important function. Intracellular transport means with the help of endocytosis, the particles are engulfed within the cell, and then all these movements within this cytoplasm is taking place with the help of actin filament. Maintenance of eukaryotic cell shape, very important because it gives the shape by the columnar, cuboidal, fusiform, polyhedral. All these shapes are according to the actin filament. Cytokinesis, that is the movements within the streaming of the cytoplasm, is also an example of actin filament. Now, these are the structure concerning about the various function of the actin filament. You see first the diagram. Look at the double helix formation of alpha actin. These are the G monomer, which are joined end to end, and they form double helix. These are actin subunit, alpha double helix, alpha F actin protein. So these are actin filament shape. What are the function performed? This is the muscle contraction, this is the relaxed form, this is a contracted form, and this is the cell cleavage, which is also a function of a actin filament. Now these are cytoplasmic streaming, mean movements of all the organelle and engulfed material within the cytoplasm. And these are amoeboid movement I have told to you during my previous slide. Now this one is the electron microscopic picture of an actin filament showing the plus end. This plus end is the growing end, which gives more addition of G monomer of actin filament to elongate this double helix of the actin molecule. This is again a picture of a electron microscopic picture visible in the cytoplasm. You see the multiple double helix formation of alpha actin. Now this is the negative end and this is the positive end. So the double helix form. Positive end is growing, whereas negative end is non-growing. What about the intermediate filament? These are the cytoskeletal components found in the cells of vertebrate animal species. Why it is called intermediate? Because its width is about 10 nanometer in between the actin and microtubule filaments. Microtubule filaments, actin and myosin filament in between, so that is why I call it intermediate filament. The microtubule has about 25 nanometer, whereas the actin filament about less 7 nanometer. As suggested by first model, all IF protein appear to have a central alpha helical rod domain that is composed of 4 alpha helical segments named 1A, 1B, 2A and 2B, separated by 3 linked regions. So it is in the form of a double helix. The alpha helical rod domain consists of 1A, 1B, 2A and 2B separated by 3 linkage proteins. Look at the picture. This is a monomer of intermediate filament. This is dimer because twisting of monomer with each other, and this is tetramer formation visible because two double helix they will unite parallel with the linker protein to form a tetramer, mean 4. Now this is the intermediate keratin filament in scanning electron microscope. Now these surrounding structures, which is the protein which is known as keratin protein, which is also an example of keratin filament of intermediate variety. What are the types of intermediate filament? Different intermediate filaments are bimentin, in general present in the mesenchymal cell. These are the primitive bimentin protein, intermediate filament, usually found in the precursor cell or the stem cell mesenchymal cells are present. Keratin is a very well known protein, again example of intermediate filament, present in the epithelial tissue, especially in the skin, certified squamous epithelium with keratinization in the surface. Neurofilaments, again the neural cell within the soma as well as the axon, they are present within these neurofilaments present within the cytoplasm of axon as well as the soma of the neuron cell. Lemon, this is the given structure support to the nuclear envelope. Now always remember, this is the point. The lemon is the type of intermediate filament not found in the cytoplasm, they present in the nuclear membrane only. Desmin play an important role in the structural mechanical support of the muscle cell. So desmin is a protein which gives the support to a structural component present in the walls of the two opposing cells, giving a type of a desmosome attaching the two neighboring cells with each other. Now, this is the electron microscope picture of a two opposing plasma membrane with the intermediate filament quite abundant blackish appearance. These are the two opposing membrane. So this is the desmosomic plaque. Plaque means a part of a where the two opposing membrane come closer and then the interdigitate fibers or of intermediate filaments give a junctional complex between the neighboring cell. So this is a important function of a desmin protein, part of the intermediate filament. Now, the third variety is the microtubules. These are the protein or hollow cylinder are about 23 nanometer in diameter. Lumen is about approximately 15 nanometer in diameter. It is a luminal microtubule tubules having a cylinder about 23 nanometer in diameter. Most commonly comprises of 30, 13, 1, 3 proto filaments in terms of the polymers of alpha and beta tubulin. Very dynamical behavior, and they are polymerized with the help of GTP. They are commonly organized by the centrosome. So briefly, the macrotubules consist of a 13 protofilaments made up of alpha and beta tubulin monomer arranged into 13 peripheral protein form a cylinder having a internal diameter about 15 nanometer, and they are formed by the organized by the centrosome present within the cytoplasm. Very dynamic because they constantly changing its length and width as well. In nine triplets, if it is present in nine triplets star shape periphery, they form the centrioles, and in nine duplets oriented about two additional microtubule wheel shape, they form cilia and flagella. When triplets at the periphery and no duplet in the center, it is the centriole. When nine duplets with having a single duplets in the center, it is the cilia and flagella. The later formation is common referred to as 9 plus 2 arrangement, wherein each duplex is connected to another by protein dynein. So always remember the tubulin A and tubulin B are interconnected with the dynein arm, as the flagella and cilia or structural component of a cell are maintained by the microtubule. They can be considered part of cytoskeleton. So always remember, dear student, that both flagella and cilia are structural component of a microtubule and microfilament. In the structure of microtubule, in eukaryotic cell, microtubules are long, hollow cylinder made up of polymerization of the alpha and beta tubulin, as I have described. These alpha and beta tubulin dimers polymerize end to end into protofilament that associated laterally to form a single microtubule, which can then be extended by the addition of more and alpha and beta dimers. So alpha and beta, they interlinked and they are joined correspondingly. When alpha, then beta, then alpha, then beta. So this is the sequence. This is the polymerization to form the microtubule. Microtubule have a distinct polarity that is critical for their biological function because they have polarity. One plus n, other is negative n. Tubulin polymerize n to n with beta subunit of one tubulin dimer contacting with the alpha subunit of the next dimer. Therefore, in a protofilament, one end will have the alpha subunit exposed while the other end will have a beta subunit exposed. These ends are designated as negative and plus end. So the arrangement is so that the alpha tubulin 13 subunit, they form a ring. This ring will attach to the next ring which is of alpha subunit, which is also 13. So this alpha and beta, these will alternate with the approximation. So one end definitely will be a beta subunit, other end, the last end, the lower end will be of alpha subunit. When alpha subunit is of the negative end, whereas beta subunit is called the plus end. The plus end is the growing end. So beta subunit end will be the plus end, and this plus end will be the growing end. So you will have to remember that they will alternate in the attachment of alpha and beta monomers in the 13 subunits to form a circle. This diagram will perhaps clear your concept that this is the cross section of a beta tubulin. Now count down, these are 13 subunits that form a ring, and this ring will alternate with the alpha tubulin ring. So this will be a plus end, this will be a negative end. When it will elongate, this end will become positive end, and ultimate the last end will be the reddish alpha tubulin will be negative end. This will be the growing end. So this is the arrangement of a microtubule alternate with the alpha and beta tubulin. This diagram is very good to explaining how arrangement of the alpha and beta tubulin will occur. Now this is the diagrammatic presentation of a cilia or flagellum. This is the 9x2 duplex formation, which are very important in the epithelial tissue, where they form the cilia as well as the flagella. One of the principal function of inner sheath radius spoke is the stabilization of the axonomy bending. Now look at a picture of this. Now this is the central duplex, alpha and beta, surrounding by the inner sheath. Outside there is the 9 duplets starting from the 1, 2, 3, 4, 5, 6, 7 and 8 and 9 in clockwise fashion. And always the alpha tubulin have a dynein arm, which are the powerful elements for the movements of this microtubule. We should have to remember it, dynein arm. You should very much remember about this. Each duplex is connected with the next link is a consonant protein, and the outer peripheral 9 duplets are connected with the central duplex by radial spokes. By the radial spoke which is connecting the inner sheath. Now this is the structure of the whole cilia and the flagellum. The relative position of alpha and beta tubulin and their dynein arm give each cross section of the axonomy a distinctive polarity clockwise orientation 1 to 2 direction. Now this is the clockwise orientation, central duplex attached with the radial spoke. This radial spoke connecting the central with the periphery, whereas the next one connects the peripheral tubule into one another, whereas the dynein arm, these dynein arms are attached on the alpha subunit, alpha tubule. So this is concerned for the movements of the flagella and cilium. Now this is the difference. Look at the picture, centromere, the microtubule and centromere, the difference. What are the difference? There is no duplex in the center. Although inner sheath is present, but there is no duplex present. Radial spoke is present. Now this is next is present, but the peripheral tubules are triplet. Although they are 9 triplet, but there is no central duplex. So this is the very much characteristic picture of a centrosome present within the cytoplasm. It is the mother component of the formation of microtubules. Now these two diagrams are the important diagrams, and these are the dynein and kinesin protein, which are the motor protein. Dynein, I have already told to you that these are the motor protein concerned for the movement of cilia, and these kinesin protein, these are also attached on the microtubule, and they are concerned for the movements of the various organelle within the cytoplasm. Now these dynein protein, these are motor protein, they are concerned for the movements within the tubule A, the alpha tubule and beta tubule, whereas this kinesin protein is meant for the movement of the various viscera within the cytoplasm. This is again a diagrammatic presentation of the same structure, dynein and the kinesin in big detail. These are coiled part, light and intermediate chain, light chain and kinesin, and this is the dynein. Now, this dynein is concerned for the movements of the alpha tubulin and beta tubulin within the microtubule, whereas these are the concerned kinesin for the movement of the various organelles within the cytoplasm. Microtubule play a function role intracellular transports with dynein and kinesin. Very important. Itself movements, cilia and flagella, and the movements of the various organelles within the cytoplasm. For example, transport organelle, mitochondria, and vesicle. Vesicle are of the lysosomal vesicle. Cross section of the cilium and flagella 9 to 2 present, quite obvious, and it is a very important function. They form axonomy of cilia and flagella. They are involved in mitotic spindle, synthesis of cell wall in plants. Mitotic spindle for centromere, dividing cell. This is again a presentation of a cilium cross section. This is 9 duplets, periphery, alpha and beta tubulin, nexon, tibial spokes, and central duplex. Now this is the diagrammatic presentation of a cilia and flagella, electron microscopic picture of the microtubule duplex and peripheral 9 duplex and a central duplex. Radial spokes, linear arm are also visible. This is the centrosome, no duplex in the center, triplets at the periphery, but 9 triplets. Now this is the microtubule and this is the flagellal formation, same structure. Same structures are visible in the flagellum. Now this is the important structure, important slide will demonstrate you the movements of the cilium and flagellum. Now look at the picture. So this is the alpha subunit of microtubule, this is beta subunit of microtubule, this is alpha and beta. The alpha attaches on the beta with the dynein arm. So this will first attach, and when attachment occurs, the ATP will provide the energy for movement. So ATP is the flash, is the battery of the movement. So therefore when attached, so it will slide past the beta tubulin like a cross bridging, pull and relax, this movement. Pulling and relaxation will move and slide over the beta tubulin, alpha unit slide over the beta tubulin, and hence it will move upward as well as bending forward. So this movement will take place, that is the forward movement, backward movement. This will all occur due to the attachment, grabbing, and then detachment of the dynein arms of the alpha subunit on the beta subunit of the microtubule. So duplets slide each other. So when sliding occurs, elongation as well as movements will take place. So cilia and flagella will bend. So this movement dependent upon the ATP molecule, which gives the ready-made energy provision to this movement. This is again a diagrammatic presentation, the ATP flash on this movement. Visible, these are dynein arm. Movement takes place forward. So tubulin A will slide over tubulin B, and when it moves, so there will be bending as well as forward movement. So it will be typical presentation of movements of cilia. So ATP molecule will provide the energy. So this is the organelle movements. Over here also the ATP molecule will play a role. So organelle is attached on the kinesin protein, which has a specific receptor for a specific organelle. Always remember, specific receptor, specific organelle, specific kinesin molecule. So these kinesin protein, when will be attached on this organelle, it will move along the alpha tube and move with the microtubule on the long elongate side, and characteristically will be moved toward the proper place of that organelle. So these two are the motor protein, dynein and kinesin. These are the function of these two proteins. Quite obvious in this diagram, in this slide, we should have to be remembered. This is the, for your interest, that is the comparison of the microtubule, intermediate filament and microtubule. So structure double helix, subunit actin, Examples are the vimentin, glial fibril, acidic protein, neurofilaments, intermediate filament, keratin. So alpha and beta tubulin is the microtubule example. Protofilament 23 diameter. So this is the comparison you will have to write down and memorize. Now this is the for academic interest of example as well as the dimension as well as the main function of all the three components of cytoskeleton. You should have to memorize this again. Thank you very much to the lecture. Next, inshallah, we further proceed through this e-learning program. Thank you very much. Allah Hafiz.